Natural cells, the immune system's first responders, are vital for the rapid detection and elimination of cancer. Photo credit MaxCyte Incorporated. While NK cells activate receptors such as CD16 that bind to monoclonal antibodies on target cells, they trigger an anti-tumor immune response called antibody-bound cellular cytotoxicity. Combining adaptive NK cell infusions with MABs is a promising potential cancer treatment. Daratumumab is a monoclonal antibody that adheres to CD38 and has clinical success in treating multiple myeloma, making it an attractive candidate for strengthening NK cell infusion. Still, because circulating NK cells show high amounts of CD38, DARA can reduce the effectiveness of this treatment combination by inducing the NK sibling link to ADCC.1. Combining DARA with adaptive transfer of CD38 knockout NK cells can potentially increase ADCC and increase NK cell permanence by reducing sibling caused by DARA. To further increase their potential, NK cells can be designed to express a high-affinity CD16 receptor that has been shown to increase ADCC and lead improved clinical outcomes too. This article showed how MaxCyte registered trademark symbol activates multiple genetic editing in NK cells. Double-regulated NK cells improved the treatment of DARA and improved anti-tumor responses in vitro and in vivo. Advantages of MaxCyte electroporation include high efficiency, minimal toxicity, and clinical scalability, which enable the rapid development of an effective cancer treatment. Purpose: this innovative research from Child's Lab has used complementary genetic engineering methods to produce NK cells that are resistant to advanced ADCC potential and sister slaughter in DARA's presence. The findings reveal that MaxCyte electroporation has minimal impact on cell survival and function, providing a safe platform for the development of cell-based cancer therapeutics. Methods: expansion. Ex vivo expansion of isolated NK cells was carried out for a week. Ex vivo expansion of isolated NK cells was carried out over a week. Transient CD16 expression (ATX) registered trademark symbol was used to transfer CD16-158V mRNA to CD38KO NK cells 7 days after initial electroporation, leading transient binary regulated NK cells. ATX was used 7 days after the initial electroporation to transfer CD16-158V mRNA to CD38 NK cells, resulting in transient binary regulated NK cells. Stable CD16 KI-CD38KO NK cells were transfused RAAV6 with an HDR template encoding the CD16-158V gene and N-terminal flag tag 30 minutes after the first electroporation. CD38 NK cells were transfused with RAAV6, an HDR template encoding the CD16-158V gene with an N-terminal flag tag 30 minutes after the first electroporation. Characterization: the designed cells were isolated and characterized using PCR and flow cytometry. The functionality of isolated NK cells was determined by an in vitro killing experiment and in murine models of multiple myeloma. High-efficiency gene knockout. MaxCyte registered trademark symbol allowed effective CD38 deletion in 92% of extended NK cells. The electroporation was gentle on cells as their vitality remained high in the days after transfusion. High-efficiency gene editing. A. Relative expression of CD38 in wild type and CD38KO NK cells. B. Anshin staining and flow cytometry were used to determine NK cell viability 8 days after electroporation. The degranulation was measured by lysosomal-associated membrane protein 1 production on NK cells in the presence of target cells and was comparable in CD38KO and CD38 weight cells. In the presence of target cells, intracellular IFNY and TNF production showed that NK cellular cytotoxicity was similar among unaltered and designed NK cells. NK cell characterization. After electroporation, the proliferative and functional properties of NK cells remain unchanged after electroporation. A. CD38KO and CD38 wait in K cell expansion 14 days after electroporation. B. Signal expression following a common culture, with target cells in CD38KO and CD38 wait in K cells was determined using flow cytometry. Rogi cells treated with rituximab were used as positive control. B. Surface marker expression in CD38KO and CD38 wait in K cells. C. The degradation was measured via CD107 expression. D. NK cytotoxicity was determined by intracellular expression of INF and TNF. MaxCyte electroporation was used to show that the expression CD16-158V could increase the effectiveness of CD38KO NK cells. Improved functionality CD38KO NK by CD16-158V expression 1 day after electroporation in various effectors, or in the present CD38KO NK cells without CD16-158V expression. The target rate cell lysis TAR treated NCI H929 cells. Under Creative Commons License Distribution 4.0 International, CC-BY 4.0 multi-level engineering stable multi-layered engineering was achieved through a combination of electroporation and AAV transduction. CRISPR-Cas9 RNP electroporation is used to silence CD38, while A.